to thank the organizers for giving me the opportunity to speak here this afternoon. What I'd like to tell you about is maybe a lesser known field of forest genomics, and I want to tell you a few vignettes of how we're using genomics in the field of forestry in practical applications to inform things like policy, tree improvement, and industrial applications. We do have genomic resources for forest trees. We actually have genome sequences for a number of different trees, and these are just a few examples. Myself, I work on conifers, and my favorite species are spruce and pine, and you can see that these guys have really big genomes. So we have a number of areas that our forestry community, our genomics community internationally has been active in taking these genomic resources and moving them along the research continuum towards application. And I'm going to tell you about 3 of these stories that we've been involved with: risk assessment, tree improvement, and traceability. And the reason why I've picked these 3 to tell you about today is because these either have direct links to the ranching sector, or we're using techniques that are very similar to what are used in the agricultural world. The species I'm going to spend the most time talking about today are lodgepole and jack pine, as well as a little bit about white spruce. All of these are keystone species in this province, so important commercially as well as to our environmental integrity. So the first story I'm going to tell you about, and this is the one I'm going to spend the most time on, is a risk assessment project. And this is where we're using genomics to help us in the risk assessment process for the mountain pine beetle outbreak. So to date, this outbreak has taken out 19 million hectares of pine forests in Western Canada alone, and that number is growing all the time. So this is a project that I've led for the past decade, where we integrate genomic techniques with ecology techniques and modeling groups to try to improve our risk assessment methods, our risk assessment tools that feed into the programs of a number of different provincial agencies and other partners, including industry partners, in policy development, spread control, forest management planning, as well as the National Forest Pest Strategy. The major thrust of this particular project is trying to understand how population dynamics of the beetle might change as we see growth from the traditional area, the historic habitat of mountain pine beetle where the outbreak first started over 15 years ago, into these novel habitats. If we're trying to predict spread and the risk of that spread, we need to know something about those dynamics. And will those dynamics be the same, or will they be different as we saw in these historic habitats? That's the driver of this particular project. And so one of the key areas where we first used genomics to influence policy and action was using genetic markers to show that mountain pine beetle had spread from its traditional host, lodgepole pine, through this area of lodgepole pine, jack pine hybrid into pure jack pine, and it did that in 2010. Now mountain pine beetle could be considered an invasive species. Even though it's native to Canada, it is now invasive in new regions. So that changes the mandate of how we deal with this beast. And where it made a difference was in the provinces east of Alberta. What we might be more interested in, mountain pine beetle, the outbreak has been slowly percolating in Jasper National Park for a number of years. The last couple of 3 years, it has exploded. So if any of you have taken vacation or driven through Jasper National Park, you might have seen lots of red trees. You might have wondered what those were. That was mountain pine beetle. This picture is in Yahatinda Park Ranch. This is quite close to where that last picture was. This is the headwaters, the Red Deer River, which, as we know, flows through to eastern Alberta. Primary source of irrigation. We know from several studies that have been carried out by others here in Alberta and in BC that there can be substantial hydrological impacts of mountain pine beetle when it takes hold in our forests, like the eastern slopes where we have really significant watersheds. That can impact on ranching, that can impact on agriculture. What we've been doing is trying to understand what that risk of spread might be, not only across northern Alberta and marching towards those other provinces, but also those eastern slopes. And one thing we've been able to do with these genomic resources is show unequivocally that tree defenses are compromised by drought. So in years of drought, mountain pine beetle is more susceptible to attack, particularly at these low attack densities. So at the leading edge of the outbreak is when we get these low attack densities. Normally, if a tree is healthy, those attacks are going to fail, or if the attacks are successful, the offspring are going to fail. But when a tree is undefended or has low defenses, which happens in years of drought because those defenses are compromised, those trees can be successfully attacked. I know this looks like maybe a map of the London Underground, but what this is patterns of gene expression, and we can compare them between lodgepole pine and jack pine, lodgepole pine under drought, not drought, and we can begin to see the networks of gene expression that are being perturbed by drought. We can see the differences between lodgepole pine and jack pine, and there are substantial differences. By comparing the water deficit situation, the drought situation, to the well-watered situation, using these genomic techniques, we've actually been able to uncover clues about how the signaling works under water deficit conditions to diminish that defense response that would be otherwise invoked by the beetle when it attacks. And so now we have direct lines of inquiry that we wouldn't have had otherwise. So others in the group have been looking at mountain pine beetle dispersal. Dispersal is the holy grail of risk assessment, understanding how beetles will fly, what affects their flight, how far they'll fly. We can also be able to tell where beetles come from based on their genomic signature. So if we're in the eastern slopes now, and we collect beetles in traps, we can tell you, did they come in from the north, from Jasper, or did they come in from the south? Are they coming in over the ice fields even, perhaps? We can tell that by their genomic signature. That tells us where the control needs to be done to protect certain areas. More importantly, we can start to get a sense of the genes that control that dispersal, because behavior influences how far beetles fly and where they can get to. And so if we can understand that, we can identify regions where beetles will fly differently than regions where they might fly further, for example. All of this is going into risk models. We're building genomics now into stand susceptibility models. These are used by the province to enact their spread control, because you cannot control every tree that gets hit by a beetle. So where are you going to spend your money? So now I want to move on to the next topic, and this is tree improvement. So breeding is a big concern, obviously, in the cattle industry. Tree breeding is also where we spend a lot of time in the forest technology industry. These are some of the traits that we breed for. My group is involved in some drought work, western gall rust work. We're also looking at mountain pine beetle resistance, and we even have a project in nitrogen use efficiency here in my obligatory cattle. Most of the time we're looking for nutrient use efficiency in marginal habitats, trying to grow trees in conditions where other things won't grow, for example. But sometimes we want to grow trees, for example, as hedgerows to protect waterways. And so in this case, we want a very high nutrient use efficiency so that these trees can remove those contaminants out of the ground. And so this is one project that I'm involved in where we phenotype these trees in order to get down here where we can use the markers linked to our trait of interest, not only for selection and breeding, but also for conservation. Last project I want to tell you about is traceability. We plant out probably 500 million seedlings a year in Canada in reforestation programs. And so those seedlings come from cones that are collected in the cut blocks prior to cutting. We need to get the seeds extracted from those cones. We need to accession those seeds. And then there are withdrawals from the seed vaults so that those seedlings can be produced in facilities like this. They get lifted, they get stored, and then they get sent out in reefer vans out to the planting site where, as everybody's already done, we go out and we plant them, right? And so following this, there's ever so many places where there could be a mix-up of the genetic stock, whether it's at the point of cone collection, whether it's the point of accessing, withdrawal, even mix-ups in greenhouses. The good news was, with this chip, we were able to verify that those seedlings, those 500,000 seedlings, were in fact from the seed lot they were intended to be, all solved with genomics. And what we'd like to do now is develop a smaller custom traceability chip. I've given you 3 vignettes of how we're using these genomic technologies in forestry, for policy, for clients, for tree improvement, ultimately all geared towards forest health, forest vigor, forest sustainability. There are a number of other applications, all of the applications listed here, with the exception of perhaps biosensors and reclamation. We have applications that have come to fruition within our international forestry community where genomics has a product out there that is being used on a regular basis to inform application. But I think where we're heading is where you guys are already moving towards, and that's precision forestry. We heard about precision medicine. We know that there are forays into precision agriculture in Canada, particularly within the crop applications, but also within livestock. This is an industry that I think is ready to get to precision forestry. And so genomics, I think, has a big role to play to getting the Canadian forest industry to precision forest management. Obviously not over the whole range of our forests, but over those high-value forests where we extract the most value. Thank you.